Hi, this is Dr. Arumugam, Assistant Professor and Head, PG and Research Department of Botany, JJ College of Arts and Science, Autonomous Pudukkottai, Tamil Nadu. In this video, we are going to see about the plant tissue culture. What we have to know, what is plant tissue culture? It is a technique of growing plants cells, that is plant cells, tissues, organs, or it may be a seed or any other parts in a sterile environment on a artificial nutrient medium is called as plant tissue culture. So in this slide we can see that what are the stages are there with a flow chart like this. If you are going to have a embryo culture or you can have a organ culture, or you can go for the callus culture, you can go for the cell culture. So each and every culture have a individual value as well as advantages as well as the uniqueness. And the history of a plant tissue culture is concerned, the first man is from German Heberland. He developed a concept of in vitro cell culture in the year 1902. The Cook in 1955, he discovered the cytokinin, for an example kinetin, and also he came to know it is potential to do that cell division. As like in 1957, the renowned scientist Dr. Cook has predicted the shoot as well as root initiation may be done by using the growth hormones with a scientist called Miller, and followed by the student of Skook, Mr. Murasik. He developed a standard protocol of the species in vitro, naming called as Murasik and Skook medium. Then we have a very big question. Said you are telling about the international scientists who are related to plant tissue culture. What about our Indian man? What about our Indian researcher? Also, we have a very good renowned scientist, renowned resource persons who have involved in the plant tissue culture. Sipra Guha Mukherjee, as well as the famous man is Maheshwari. Both in 1966, they developed the production of haploid plants from the Datura inoxa. It is a totally haploid plant. Previously, the agriculture scientists used to depend on the emasculation. After that, this invention by those two persons, the agriculture scientist has switched on to the plant tissue culture for production of haploid plants. And what are the fundamental principles are there in a plant tissue culture? Means, first, the concept or principle is called totipotency. The totipotency is nothing but the ability of a particular or individual plant cell can do or can generate into a new whole plant. So a particular or a individual cell can create a whole organism is the phenomena is called as totipotency. And also another one is plasticity. The plasticity is nothing but it is ability of the plant to alter their metabolism, growth, as well as the development to the best suit their environment, because the plasticity is essential for the plant, for their metabolic activity, for their growth as well as development by adopting their environment, and has to emphasize or has to do that these three major functions. As like this image is coming to say about a plant cell, it is isolated as an from the plant, that organ called as explant. The explant, when you are going to do that in an artificial nutrient medium, that medium which is develop the new cells by the process of mitosis, cell differentiation, cell elongation, and etc etc. With these phenomena, or with this protocol, the plant can, the individual isolated explant or plant cell can give a new individual plant. So a leaf which is excisted, which is taken from a mother plant, that plant is going to give a cell, that cell is going to produce a new individual as like mother's plant. So this is called clonal propagation. In that, when you are going to grow a cell in artificial medium, it has to undergo so many processes. It has to differentiate and redifferentiate and de-differentiate. So this process has to do that to form a new individual or new organism. Then last slide I have mentioned 3 or 4 times about the explant. The explant is nothing but the excised portion of a mother plant. Generally it is called as multicellular tissue fragmentation, called as explant. It is obtained from the living plant or mother plant. The plants may be originated from the wide range of plant tissues such as leaf, stem, it may be a root, or it may be a petiole, or it may be a hypocotyl or cotyledon, embryo, or meristem, and etc etc. So the illustration come to say you is the system of the plant is consists of so many organs. It starts from the apical meristem to root apical meristem as well as shoot apical meristem. So the branches which is connected to the main trunk or main branch, it have a node or internode or like petiole or bracteolate or leaf bud and leaf blade and nerves, so on those things. You can take whatever you want to produce a new plant. That phenomena what I said earlier is totipotency. And okay sir, these are the good techniques are there. We have to know the advantages of the technique is called the plant tissue cultures advantages. The first and foremost is the plant tissue production is reliable as well as constant. So we can do the production of large level of the plants what you want or what you desire or what your interest. It should be very reliable, because what I told you this, because the single cell can copy, can do the regeneration of a new individual as like mother cell. So we can have the reliability and consistent. So we have the consistent of the production plants, and also the multiplication rate also the very high. If you are going to propagate by the seed propagation or the vegetative propagation like cuttings, layering, craftings, we can have some minimum level or we can have certain ratio. But when you are going to do that by using the plant tissue culture system, we can produce the multiplication of the plant in a high scale or large scale level. The plant produced via tissue culture are usually true to type, as it is cloned or the original from mother cells, and it may be uniform, and it can be produced the mature plant very quickly, And major and very important thing is we can produce the disease resistant plants by using the meristem tissue culture. And also the high rate of fecundity is obtained from the plant tissue culture. Okay, sir, you have mentioned, dear friends, anyone have its advantages means also it may have some disadvantages. When you look upon the advantages, also you have to look upon the disadvantages also. Then only we can do the technique or experiment in better way. This plant tissue culture or technique or experiment which involved a tissue culture laboratory are somewhat expensive than the conventional method of tissue culture, conventional method of propagation. The handling of highly trained people, because it has to be aseptic condition. So the people who are underwent this so much of training, they can do tissue culture in a better way, and also they can do the observation very carefully. All the plants are genetically similar. There is reduction is genetical diversity. Because of this evolution or the genetic diversity, we need some improvement. By the clonal propagation or getting the identity or the similarity of the same like mother cell, so we don't have a genetic diversity. If precautions are not taken, the whole stock may be contaminated or infected. What is this? If a trained person is not utilized for the purpose of tissue culture technique production, if any person is getting the somewhat not precautionary, we may lose the whole stock because of the contamination or any infection of a fungus or whatever it, bacteria, whatever it is. Also, the plant tissue culture is a complex procedure. It has a varied procedure to require the special as well as careful observations. What are the techniques, what are the steps, or what are the complex procedures are there, we can have it in the next two coming slides. After coming to this knowledge, we have framed or we have a certain defined protocol, procedure, or steps involved in the micropropagation technique. First, we have to clean and prepare the glasswares for plant tissue culture. The glassware which are going to use for the plant tissue culture technique starts from the explant preparation to hardening. So we should maintain some glasswares, test tubes, beakers, culture tubes, and so on. So it should be cleaned very hygienic manner. Simultaneously, you have to prepare the nutrient medium which is going to supply the nutrients or food for the growth of the cell or any explant. And also you have to do the sterilization explants. Not only explants, we have to sterilize the medium and surface area where we are going to work. Inoculation chamber, culture room, and so on. The inoculation of the aseptic explants into the nutrient medium, because after the sterilization of the explant, it has to be transferred in a nutrient medium. That process is called inoculation. The next one is proliferation of the shoot on a multiplication medium. So we have to standardize the hormones for initiation or multiplication or elongation of the shoots in a different ratios of the hormones. The transfer of the shoots for the subculture is to be followed for getting the large number of or better growth of the individual clone. The rooting and hardening of the plantlet is very essential, as like a child. We have to take care of these younglings for getting rooting. After getting the better rooting or elongation or multiplication of roots, we should do the hardening of the plantlets by the process. And also we should have some field experiments after the hardening process. By the field trials or after the transfer of the plantlets into the fields, if our micropropagated or plant tissue cultured plantlet is grown good, it is survived nice, means our protocol, our procedure, our experiment is good. Until otherwise, we have to repeat that experiment from the first step itself. Okay. Okay, sir, good. Then I want to start, or I want to know the plant tissue culture laboratory, what would be that? So need of low cost laboratory. Sir, I have one idea. I wish to start one low cost laboratory for the plant tissue culture. What I supposed to do that? The options should be a lower cost of production without compromising the quality of the micropropagation of the plants. So we should not any compromise in certain things. The micropropagation technique is too much of costly, not too much of costly, but we should invest certain amount. Certain amount we have to invest. Then only we can have a good laboratory. The potential increase in the interest in the agricultural crop production is increased by giving the low cost laboratory. Low cost tissue culture technology will stay a high priority in agriculture, because in agriculture means we have to perform the agriculture or we have to conduct the agriculture with the low cost effective. If agriculture is getting the high cost, there is no meaning of agriculture to the farmers or poor peoples. So as you remember this, you have to give the priority in agriculture, horticulture, forestry as well as floriculture of the many developing countries for production of this suitable priced high quality planting material. Okay. So foremost, the forthcoming slides we are going to see about the general requirements for setting the low cost laboratory. Okay, sir, I want to initiate a low cost laboratory for the agriculture sector, or it may be horticulture or in a forestry, whatever you want. So this is an illustration or a drawing of a low cost plant tissue culture. So we have the entry, the right side we have a chemical storeroom, then analytical instruments, then photography. In left side we should maintain the very big benches or any tables for giving some experiments or for collection or preparation of explants or media preparation and other things. So we can maintain a 2 to 3 benches or desk or working bench for performing such activities. And washing room you should have the separate, because we are going to wash a fresh or any contaminated cultures in a washing room. So the washing room should be connected with the culture room or main hall, or it is a working area, and also sterilization area. Then only it will be feasible to work or wash the things which are urgently required. And we should enter the airlock, because the airlock, the floor is there. We should have some cleaning of dust particle on our body. We should have some airlock. And also in the dissection room we have to maintain, because we should perform some anatomical or histochemical techniques after the tissue culture. And also in tissue culture rack and tissue culture cells, we should have some inoculation chamber, and also from the inoculation chamber we have to go for the culture rooms. Okay sir, I want to start a low cost plant tissue culture laboratory. What about the location where I can start? Which location may be better for starting a new laboratory? The location may be confirmed to the land use plan of area. The building should be located away from the source of the contaminated, such as a soil mixing area, storage, and municipal waste disposing area, and other things. The site shall be accessible to the service roads, water supply, and also electric lines. And also the service load should be connected with the loading and unloading the materials what we are preparing or what we are going to purchase. And the site shall be well trained. The structural requirements. Structural requirements is very essential, because this is a one time investment. We can't reconstruct the of the structural plan. The roof structure should be trained the timber or steel with anti-rust paints. The roof vents which provided shall be properly screened. Ceiling should be at least 2.4 meter from the finished floor line, as well as painted with latex paint, and should be prevented from the dust. The wall shall be concreted. It has smooth finished like putty like this. Properly painted with antifungal epoxy paints. Nowadays we have a antifungal or antibacterial or antimicrobial paints are there. We can perform like this. And the window shall be properly installed with 15 mesh. That is a number is there. There is a number is 16. The mesh level. So we have to screen at least 1 meter from the finishing floor of line. The main doorways shall be at least 1.5 meter wide and moisture and also rust resistant material. The floor shall be concreted, smooth, non-skid. Slope should be 2% to 4% towards the drainage. And the basic facilities of the laboratory is concerned one room with computer with locker for the keeping brawn. The working place for the explant preparation, washing room, sterilization room, media preparation room, inoculation room, incubation, that is culture room, and skilled labors. These are the facilities we require for performing plant tissue culture. This is the washing room. We have a very good, whether we can have a granite as in a stainless steel system, whatever you want. So there should be a zinc, the maybe the size 45 centimeter into 60 centimeter, like 1.5 to 2 feet. The rooms it would be provided with the buckets, detergents, mugs, storaging areas, with water line, with the electricity. It should be located nearly the sterilization as well as media preparation rooms. And in sterilization room, the sterilization should be have a one autoclave. It may be a vertical or a horizontal, or a cooker with well ventilated exhaust fan. Also having the tools for supporting while placing the media bottles in autoclave as well as cooker for sterilization process. The bottle should be sterilized at 121 degrees Celsius at 15 lps for an hour or 20 minutes. It may be depends upon the whether you are going to contaminate or you are going to sterilize. That depends upon the duration will be differ. It contain the wrapping paper to wrap and the rubber to tie the inoculating the dish for the sterilization process. The media preparation, so we have a very big media preparation lecture in separately, but there should be a room separately. We should have some space and we should have some racks, shelves for maintaining um, chemicals which are required for the preparation of the medium, and all there should be provision of fridge for maintaining vitamins, hormones, and stocks. An inoculation chamber is nothing but it is where we are going to inoculate our culture initiating stage. There should be a laminar airflow chamber, and also it has to be air conditioned. It should be where we have to maintain the shelves or racks that forceps, scalpels, sterile bottles, and sterile dishes, and the sprit lamps, cottons, and what is that gas connection should be provided. The incubation room is very essential where we are going to culture the area, room or chamber should be very aseptic condition. It should be maintained air conditioned. It should be protected from the pest or any insects, and also we have to maintain the lights, or we have to provide the light like 200 lux to 600 lux. We can otherwise we can maintain the 1000 lux for the better growth, and also temperature should be maintained from the 25 degrees Celsius to 27 degrees Celsius, 25 plus or minus 2 degrees Celsius. And photo periodism has to be given, like 16 hours or 8-hour dark, and 16 hours we have to provide the lights. The light unit is 1000 lux we have to provide. This is the instrument room. These are the instruments which are required for performing some experiments during the culture or after the culture. But that is washing machines or autoclaves, the freezer, rotator shaker, then the multi-purpose magnetic stirrer, then weighing balance, a thermometer, and microscope, and whatever you want, you have to be there. The magnetic stirrer, digital pH meter, laminar flow chamber, UV chamber, air condition with the stabilizer, and other things. The glasswares. These are the glasswares which are required to perform. The experiment of plant tissue culture, it starts from the bottle, petri dish, purette, funnel, beakers, pipettes, conical flasks, culture tubes, measuring jars, then spatula, cowel pail, forceps, wrapping papers, buckets, clothes, and etc. 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 So, it is very essential when you are going to start a plant tissue culture laboratory. We should maintain the standard practical manual for the plant tissue culture. What I am suggesting is the plant tissue culture by the Bochwani and Razdan, a very good book, and also the plant tissue culture manual by Lindsay, and also the plant tissue culture, the methods in the plant tissue culture by Yuk Kumar. At least three books we should maintain. And if you want to start a low cost of a plant tissue culture laboratory, it will cost around the general chemical cost. The glassware is 15,000. All chemicals we can purchase in 25,000, and the aluminum racks and everything we can purchase for the culture in 10,000. And other requirements for the laboratory, it may come other things like come consumables and other things. It can have contingents like 25,000. Instrument may be water purifier system, electronic balance, pH meter, hot plate, refrigerator, laminar flow chamber, autoclave, computer. It will cost like this.